Hello, folks. I'm Katie.
I'm Vinny, and this is Learn Real Good.
Ah, it's back.
We did it.
It's back. We're back.
We're back.
It's good to be back in the chairs. That's what I was trying to say.
Back in the saddle.
Yes. We only sit in these chairs for podcasts.
Is that true?
You know what? So, truth, behind the curtain, we're in
Yeah.
our dining room.
Correct.
You know what? We don't use it that much anymore.
The dining room?
Recently. Yeah.
No. It's become an office, like most people during the pandemic.
Yeah.
Your dining table has become like an office, secondary office table.
Yeah, it's where we do office work. It's where we do board
Hobbies
games
and
hobbies,
and we have
podcasts.
guests. Eating, not so much.
Not so much.
We just put the food on a plate on the floor and go to town.
We've taken to, TV dinners.
Eating dinner in front of the TV.
Yeah, as a family.
It's a family activity.
Instead of talking to each other.
No. Best to be avoided.
You can't get in fights that way.
Yeah, that's right. If you don't talk, you can't fight. That sounds really healthy.
We've never been happier or further apart from one another.
That's right.
Well, this is Learn Real Good.
Yes.
What
is it?
It's a podcast.
Great.
You're a co-host, Vinny.
Whoa.
And each week we come in here, we chit-chat, we share facts,
Yes,
and then the marquee part of the show.
Yes.
The reason we're here today, the biz marquee of the show, is that we have a guest who's going to talk about the research, and I'm super excited.
I love talking to our guests.
Yeah, it's very exciting, right? I learned so much on this show. I guess that's the point, and why learn is in the title.
Hopefully our audience listens and learns stuff too.
Not just us.
I mean, mostly us. If you're out there learning stuff, that's your own business.
Well, it's really funny because, you'd think that, people outside of academia might think that people with PhDs, know everything, at least in their sort of arm
field
of science. Yeah. But it's not really true, right? The further you go, the more smaller, the more you know about something very narrow.
Yeah. First, you study the fish. Then eventually you end up studying the fin. And then you end up studying the microbiota that live on the fin.
Yeah. And then you're like...
And then you specify one of them.
Right.
And then you just know that.
Yeah. And then next thing you know, you don't know more
someone
than
says,
one
what is that
fish.
Tree? And you're like, I have no idea.
Well, that's the thing. That happens to be all the time as a biologist. People assume I know
What's
everything.
this flower? And you're like,
Nothing.
Nothing.
I never took a botany class. But
that's okay.
You know what? No one's perfect.
No one's perfect.
And we're here to say that.
We don't need to be. And we are prime examples.
Yes, happy to be
not perfect.
The examples of imperfection. Embrace it. Failure is a gift.
Failure is a gift.
Right? Okay. Well, it's time to share facts.
Science, facts.
The unofficial theme song.
That's right.
To our facts. Who goes first?
I'll go first.
Okay.
I love sharing facts.
Let's do it.
All right. Katie, how do you make tea?
Well, I don't know that I do it correctly. And you know what's interesting? I hope the mic picked that up because I just swallowed a gulp of tea. That's not a lie.
What's the first step generally when you're making tea?
Okay, are you talking about making a cup of tea or creating the tea?
Making a cup of tea.
If you're making
a cup of tea at home,
I would be embarrassed.
What's the first thing you do?
I want to get this right.
There's no right answer.
Imperfection. I boil the water. Okay, I'll admit it.
You admit it.
Okay,
Guilty.
Here I am. Guilty.
Perfect. That's the right answer.
Gosh, I was worried.
There was no right answer, but that is what I wanted to talk about, boiling water today.
Oh, great. Okay, we haven't even gotten,
Yeah, we haven't even gotten to the fact. I'm just doing all the setup. Boiling water.
Oh, I know.
You know what this is?
I know this one.
I want to hear this. I want to hear this. Tell me.
I read this.
Yeah.
Oh, crap. Okay, so people are looking at how to boil water more efficiently.
Yes.
And they found something to do with the surface and the vessel being used for boiling be dimpled.
Yes. It's actually 3 things, but being dimpled is one of them.
Okay, that's as far as I read. I got bored.
Boiling water, you're allowed to get bored halfway through an article. Boiling water is a huge process. It's very common. People do it in the home, but as an industrial process, it's used all the time, especially in power generation. Most power generation is just making steam. The Industrial Revolution, right? It was steam engines. All just boil some water. That steam rises and creates pressure, and it goes to do work in whatever machine you put it in, right? All those trains with locomotives, they have coal on them. What does the coal do?
Make steam.
You boil. All you do is a boiler. You just take water, you boil it. It's the machine that everything hydro dam.
They're all teapots.
You just
right?
have
They're
water
all just
move
teapots.
making a turbine. All of these machines is energy generation. Nuclear power, you just boil some water, you make some steam, it turns a turbine, boom, you got power.
Got it.
So boiling water is a very, very important process. And we're totally ignoring all of the industrial chemistry that goes into like boiling water for certain processes and temperature regulation and whatnot. Boiling water is important.
Got it.
So
Convinced.
Making it more efficient becomes pretty important because we're all very power conscious these days. We need to do more with less. So anything that can shave even 10%, 5% off of the energy consumed of us boiling water, that's actually quite a bit of energy on like a global scale.
Sure.
Both on a personal residential use and on a commercial use. So anything we can do to increase the efficiency of boiling water is great.
Okay, I'm convinced.
And so the way these researchers at MIT have improved the efficiency is by changing the surface. So like if you think of the kettle,
yeah.
you have the bottom surface that gets really hot, all the water boils, great.
It's really all bottom, isn't it?
Well, the heat rises, right? So if you
Sure.
put a kettle with a heating element on the top, it's a real waste of energy. Half the heat will go up through the top of the kettle. So you put it at the bottom,
Sure.
heat rises. Now,
Gotcha.
what happens is as the water makes contact with this hot surface, it creates this layer of vapor. When you see the bubbles come up from a boiling pot of water or a kettle. That layer of vapor acts as an insulator to the water. So that heat is not being transferred to the water until the bubble rises. So what you want to do
Maximize bubbles.
is minimize the bubbles that are...
Wait, what?
So if you have all of the bubbles, at the bottom of the pot, it creates this layer of gas, this thin layer of vapor that acts as like an insulating barrier. Like goose feathers are full of air, and that's what acts as an insulator.
You want freely moving bubbles.
Yes, so you don't want those bubbles to like kind of stick to surfaces,
yes,
right?
yes.
Water tension and all that stuff.
Okay.
So what you do is you create dimples. That's one thing. But they did three things actually. So they created dimples that were on the scale of millimeters. They put little posts, little pillars in the bottom of the thing as well, so that it creates like an even more bumpy surface, so that the bubbles climb up and go up out the top.
Yeah.
And then they do one more thing. They added like little nanometer scale ridges to create even more surface area.
Interesting.
So
surface area on which to make bubbles?
Yes.
Okay.
Yeah. Well, the more area you have in contact with the water, the more heat can get connected.
Okay.
So you have these dimples, you have these posts, and you have these ridges and creases. All three of these things combine to improve the contact of the heating element with the actual liquid water itself.
So how does this transfer in terms of time to boil? Do we know?
So if we're just looking at time to boil, I think they were optimizing wasn't time to boil, but energy used to boil water.
Right.
So it may take longer, but I don't think it does. If it's more efficient, it probably goes faster too.
Now, I got a wild idea.
Yeah, I'm here.
If bubbles are, such a big deal in boiling water,
correct.
why aren't we boiling our soda stream soda water? Would soda water
I'm listening.
Would soda water
I'm listening.
boil faster because it's got bubbles everywhere?
Well, if you have bubbles in something, right? We were trying to eliminate bubbles between the heating element and the liquid.
But bubbles moving through the water bring the heat around, right?
I think what a bubble is vapor.
Oh, I see.
And so it just rises up through the water and escapes.
I'm really hung up on the bubbles.
Yeah. Yeah.
If we boil bubble bath,
yes, I'm listening.
Will it be delicious?
I wouldn't recommend drinking it even at room temperature.
Interesting. Well, I mean, it's about time because teapots are one of those things like umbrellas that I feel like we really haven't put a lot of. Someone made like a very basic model, and we've all been like, it does the thing, and it's not perfect.
Right.
It could be optimized, and we use it so much. You'd think that
someone is working on optimizing.
And the answer is they are.
They have.
They are. They have. They will.
Will.
Will they? Won't they?
They will.
They did.
They did.
Great fact.
Thank you.
Thank you for sharing that.
All right, Katie. I'd love to hear yours.
It's a good one.
All right.
It's a good story. Okay. So we recently went camping.
We did.
This past summer.
Yes.
And one of the many adorable creatures we saw was a vole.
Yes. Like a mouse.
Like but we actually saw a vole as well. We saw a mouse, but we also saw a vole outside.
They were labeled correctly.
Yes, thankfully. They had little, like cheeses at a deli counter. They had a little stick. That's gross. Gross. Well, this is going to get grosser. It's voles. Little tiny mouse-like mammals. Yes. Okay. Voles and their enemies are sort of the subject of the story. Their enemies being shrikes or butcher birds.
Oh.
And they have earned such a
like
name.
Falcons?
No, they're birds that are brutal, thus the nickname butcher birds. So these butcher birds, what they do is they are known for hunting their prey, like voles, by perching themselves on like tougher tufts of grass in a field. Typically, they like to stand on something called bunch grass. So you can picture like normal lawn grass, and all of a sudden there's this like tough, higher area of bunch grass.
Like wheat.
Yeah, and that's where the butcher bird would stand and sort of survey the area looking for their prey.
So they're not very heavy.
I guess not. Well, you're picturing wheat. That's your choice.
Yeah.
I mean, I'm not saying they're standing on a single blade.
Oh. They're standing on a bunch of blades.
Yeah, so a bunch of blades.
Of tough grass.
Yes.
Okay, got it.
Okay. Again, we're getting hung up on the wrong things today.
I just want to try to get a sense of how big are these birds?
I don't know. It doesn't matter.
All right, they're shrikes. Got it.
Bigger than a kettle. Okay, so what they do, and why they're called butcher birds, is that they will catch their prey, like a vole, and then throw them, impale them on something like a thorn or something, and just leave them to like die and sit there and rot, and then come back to eat them, or use the stench of their rotting prey to attract mates. Hot.
Whoa.
Yeah.
Really?
Yeah.
That's fascinating.
Now, you might be thinking, the poor voles, they're so small and stupid. Because, well, little mammals tend to be kind of dumb.
Sure.
But they fight back. And the way they do it, or at least try to protect themselves from predators.
Armor.
Is they've built tiny swords and shields.
And they ride other smaller voles like horses.
Yes. You read this article.
I read the article.
That's not true.
Oh.
What they do is if we spend so much time on this grass, if they need this grass to perch on, the voles cut it down.
Really?
So they're actually ecosystem engineers. Ecosystem engineers just refers to sort of a role that organisms can play in their environment where they change the structure of the habitat.
Like a beaver.
Like a beaver, great example of an ecosystem engineer. But this is actually the first time scientists have documented a species changing its environment to decrease its own predation risk. So it works. So they cut down this bunch grass.
Right.
And then the scientists from this article showed that it decreased the number of incidences.
That's a good idea, though.
Strikes. Yeah.
It works.
Great. And they even found that if the scientists put like netting around the bunch grass.
Well, whose side are they on?
It's an experiment.
Oh.
So they put netting around the bunch grass that excluded the birds. So the birds couldn't perch there and hunt. But the voles could still cut it down. The voles didn't. So they were able to see that.
Whoa.
Yeah, they could change their behavior and be like, well, I might as well save energy because it costs energy to cut this stuff down. They don't eat it.
Right.
So they're just investing energy to cut down potential predator
threats.
habitat. Yeah.
Wow.
Yeah.
That's
cool.
wild.
Cool, right?
But then the shrikes are going to start like, well, we're going to seed these. They're going to start growing them. Counter engineering.
Well, I don't know much about shrikes, but if they're anything like...
They're huge planters.
If they're anything like ravens and crows, then they'll learn. Crows are crazy smart.
Yeah. We're going to have a crow as a guest coming up, one of the STEM students.
I'm intimidated because I think they're smarter than us.
They're smarter than both of us.
But I worked with birds. We were looking at...
In grad school?
As an undergrad, actually, before
studying,
grad school.
though.
Yeah.
Not as peers.
No, not as peers. My PI was a raven, though.
All right.
But crows were a big predator of the bird species we were studying. And they were so smart, they would know to follow us. They knew our routines, and they would follow where we would go, and then they would eat the birds.
Because you were studying the birds, and they knew you were studying the birds, so where the people went,
correct,
food would be.
So we had like cameras set up, so when we'd be out in the field adjusting our cameras, the crows would go and eat the birds. So we would have to set up decoy cameras. We had fake, made with like toilet paper rolls and sticks. We had fake cameras.
And a fake host and a fake interviewer.
We'd pretend to check them.
It's like a just for laugh gags for crows.
100%. They were still smart though. And when we found a nest, we had to sort of downplay it, and we had like a complicated flag ribbon way of indicating where the nest is.
Wow, that the crows couldn't decode.
Yet.
They'll figure it out.
They'll figure it out. We have faith in you, crows.
You
anyway,
got this.
Wow,
that's really cool.
Neat, right?
Wow.
So voles, not as ding-dongy stupid as we thought, or at least
I never thought they were stupid, so really.
You're pandering. You're pandering to the
The
voles.
Well, the voles make up a huge part of our listening audience.
All right, Vinny. It's enough from us.
That's enough. No one wants to hear more.
It's enough, right?
I want to hear from our guest, Katie.
We're going to. I have the distinct honor of introducing our guest this week, Ghufran Rafique. Ghufran is currently a PhD candidate in the Sleeman and Perepitchka labs in the Department of Chemistry at McGill University. His work focuses on combining DNA with non-DNA molecules to create hybrid polymers for use in nanostructures and sensors. He enjoys reading and learning about the history and philosophy of science and about religion, avoiding most fads, joy of missing out, JOMO, and occasionally interning as a squirrel paparazzo. Please join me in welcoming Ghufran to the show.
Hey,
Ghufran, come on down.
Hey, Bismillahirrahmanirrahim. Thank you for inviting me. It's a great pleasure.
Thanks for joining us. Thanks for not missing out on this podcast. This is really exciting.
Yeah.
Again, we've had a few episodes this season where we're like, we don't know anything about that. So that's going to be the theme of this show.
That's most of the episodes. We don't know anything about that should have been the alternate title.
Yes. Chemistry. Wow.
Yeah. So combining DNA with non-DNA, that's cool. Tell us a little bit about that.
So whenever I tell people that I work with DNA, most laymen automatically assume that I am a biologist. And that's what I used to think as well before I started my PhD. But I basically work on using DNA as a material, so as a polymer itself. And the idea was originally conceived of in the 1980s by a scientist named Nadine Seaman, who passed away last year. And he thought of using DNA as a nano building material to construct with it nanostructures that could then be used to position proteins precisely. That was his original idea. And the reason we're able to do this is because DNA has the specific sequence of nucleotides.
Mm-hmm.
And within the nucleotides, you have these bases A, T, G, and C. And so they're complementary to each other in pairs. And so if you have a particular sequence, you automatically program within it information for the complementary sequence. So that can then come together and bind with it in a reversible manner. And so if instead of, the double helix is something that we're all familiar with. So instead of the two strands within a double helix, you bring in a third strand that partly is complementary to the first one, partly to the second one. You can basically make a three-way junction now. And then if you increase the number of strands, then you can basically create more junctions. And then when you have junctions, you have these small building blocks that you can then use to create a larger structure. So then in 2006, there was a scientist at Caltech. His name is Paul Rothmans, and he designed this idea of DNA origami, where he took a really long DNA strand that was, I think, bacterial DNA. And then by using short strands of DNA, he folded the large strand like origami. But he used the small strands that were complementary to different parts and portions of the sequence of the large strand into a nanostructure. So using this technique, he was able to make smiley faces at the nanoscale that could then be visualized using experimental techniques. So that's how you can use DNA as a nanomaterial. It's an old concept, but it's a really growing and exciting.
Whoa. Okay. So let's break this down. So I know a bit about DNA. I'm going to leave it at a bit. But I know, like, in my body, Ghufran, like, I've got my double helix kicking around in my cell. And if I want to duplicate it, right, we've got enzymes that are going to pull those puppies apart. And then each of those separate single strands are used as a blueprint to build identical second strands. And boom, I've got two double helices now. When you're sort of working on this in the lab, are you utilizing enzymes to build DNA, or do you have like all your Lego blocks pre-built and it's a matter of just making them stick together to build bigger stuff?
So we actually use the latter, which is we use a DNA synthesizer with a computer that basically has lots of valves and it has these ports, and we create these DNA. We actually build this DNA one block at a time, so one amidites at a time to create these nucleotides one after the other. But typically on the synthesizer, we don't go beyond 100 nucleotides, so 100 bases of single strand. Because beyond that, the yields start going down pretty fast. The yield of each individual coupling is very high. It's about 99.9%. But then as you grow the strand, the efficiencies get multiplied. So there is somewhat of a decrease. It's not the most efficient process because we use a large excess of reagents, but because we do it on a very small scale, it's let's say fiscally viable. for us to synthesize that.
That's amazing. And
yeah,
I'm picturing like basically it's like the computer's like put in the blue block, put in the red block, put in the green block, like Lego bits. Obviously, it's more complicated than that. So how many different inputs do you have options to put in when you're printing these? Like, is it like 3 ingredients or 100 ingredients?
So fundamentally, we have the 4 amidites associated with the 4 bases for the nucleotides. But then here's the interesting thing. Our lab actually also adds non-DNA based structures. Mm-hmm. So, and then we can also change the chemical composition as well. So instead of making DNA, which normally has oxygen and phosphorus bonds in it, we can replace the oxygen on the phosphorus with a sulfur. And so now we have a sulfurized DNA, which is actually a bit more stable biologically. And some of the students in our lab are actually using that to make DNA-based nanostructures for nanomedicine. Whoa. So not only can we change the units that are added, the blocks that are added one at a time, but also the chemical composition of the links, which are phosphate diesters within the blocks. So we can make them thioesters of sulfur instead of oxygen.
So you're building this one letter at a time. Do you reuse words often? Are you like GATC, GATC? Or is it randomized? Or is there some sort of complicated blueprint of the letter sequences that's going to dictate a shape?
Is there some method to the madness?
Yeah, so if we're doing something for the first time, let's say we're making a strand of DNA with non-DNA building blocks, and we're not fussed about the specific sequence, we use just an arbitrary sequence, which is a 9-teamer that by now I've memorized. I can recite it in my sleep. But if you're looking for a specific functionality, let's say one of the projects I'm working on requires that I have a specific number of thymines in a sequence. Mm-hmm. We're trying to explore what happens if you move them around. So then you have to basically input the sequence according to that.
And I guess the question for me is, are you working towards a goal, or are you just like, let's try this out, and what happens if we put this here? Or are you like trying to like, well, how do I make a model Eiffel Tower out of DNA bits?
Yeah, so ultimately, the goal of my project is to create these hybrid DNA polymers that have DNA in 1 or 2 parts, and then they also have these functional non-DNA based conducting or semiconducting monomers or parts that are basically part of the chain itself. So then the structure can fold itself and form nanostructures. Before I started working on the functional, the semiconducting and the conducting monomers, I was also working on, I'm still working on adding basically just plain building blocks that are non-DNA, and then looking at how the polymer itself comes together in water to create nanostructures.
Now, I understand, Ghufran, with the rules of complementarity in DNA. So that's just so just for people who are less familiar, there's sort of 4 basic building blocks, letters of DNA, GATC, and they pair up specifically. A's and T's like to pair up, G's and C's like to pair up. So it will stick to itself and can build 3D structures, at least when it's a single strand. So why is it necessary to have these non-DNA things? Is it that you can control how it folds better, or does it fold stickier?
So it's basically, think of it as sort of a toolbox, right? So when you have just DNA with these A and T and G's and C's that are complementary to each other, you can only use the rules of complementarity of DNA to fold the structure. But then there's also, in terms of polymer chemistry, there's also something called long-range interactions. So these AT, GC interactions within the DNA, they're short-range. So the diameter of the double helix is about 2 nanometers, right? So they don't extend throughout the solution. But what you can do is you can use non-DNA based interactions that are longer range interactions. You're introducing these non-DNA building blocks, and that allow you to basically expand the toolbox of intermolecular interactions between the polymers themselves. So the most basic example to explain this concept, which is called phase separation, is the soap micelle. Right? There's a lot of focus on using soap in order to, or hand sanitizer in order to ward off the virus, and that's because of the way the soap works, right? So soap is basically, it has this long carbon chain with a polar head, and so the polar head likes water, so it's hydrophilic, and the non-polar tail, which is made of carbon atoms, hates water, so it's hydrophobic. So when you put this structure in water, it's not a polymer, but it's a molecule. it assembles itself to stabilize itself with the hydrophobic chains going on the inside and the polar head on the outside to interact with the water. And so if we design a DNA-based polymer in the same way with something that is hydrophilic, which DNA is, with a portion of monomers or blocks within the sequence that are hydrophobic, then again, you will form these nanostructures with the hydrophobic stuff on the inside away from the water and the hydrophilic DNA on the outside. And so you've used basically, you haven't used the DNA interactions to build this nanostructure. You've simply used basic hydrophilic, hydrophobic interactions to make the nanostructure. And then this frees up the DNA, which is still part of the nanostructure. It's on the outside, it's waving about for additional interactions. So you can build on this nanostructure.
Wow. And so you're making these like 3D pieces, basically.
Yeah, exactly. Yeah.
And is one of the goals or possibly one of the applications like you can build like a little nanostructure machine out of these interacting pieces?
Yeah, you can. One of the projects that some of the students in our lab are working on is to create these nanostructures that, for instance, are spherical. That's the more straightforward geometry. And using the DNA itself as a therapeutic piece. So you have the mRNA vaccines, which have the messenger RNA within them, which is an oligonucleotide therapeutic. It falls within that category. But if you have something that is, instead of an RNA or an mRNA, if you have something that's DNA, but it's still therapeutic, and it can block the production of proteins in the body that are related to disease, then you can basically create these nanostructures as therapeutic vehicles. So they have DNA on the outside that is therapeutic, and when it's internalized into the cell, then that DNA portion is used to basically inhibit, for instance, the synthesis of these proteins. But my project, has, instead of just these carbon chains, for instance, ultimately it looks at how to integrate electronically active blocks so that when the structure folds, it retains this electronic functionality on the inside. And then it has this biological or short-range chemical functionality on the outside in the form of the DNA.
And I guess my question, what does that mean, electronic activity?
So if you have a semiconducting block or a conducting block, in the context of my project, we want to insert these inside the cell membranes so that we can use this as a sensor to integrate the outside of the cell with sort of a sensing device with the inside of the cell. And so there is a chemical activity that, for instance, generates a piece of DNA, like associated with a virus, for instance, that is then going to bind with the DNA that is part of the polymer that is embedded in the cell membrane. Then the structure itself could reposition within the membrane. And this would then affect the way the electron transfer, for instance, or the exciton transfer through the stack of the conducting blocks or the semiconducting blocks is taking place from the inside to the outside. But that's like the ultimate goal,
right?
If it works.
And then you detect that change.
Yeah, exactly. We detect that change on the outside.
Wow, amazing. So they're basically, like little sensors on a DNA level.
Yeah, exactly. The challenge is how do we integrate something that's not inherently biological, so something that's like a conducting block with DNA. Right. So there are chemical challenges to overcome.
Of course.
Yeah.
It can't be that easy.
No, so it's the overall project is actually a team effort with 3 different groups. So the conducting molecules are synthesized and tested. My job is integrating them to make them biologically stable with the help of DNA. And then the sensing is actually done by another group, the third group that's part of it.
Wow.
Yeah.
So one thing that comes to mind, Ghufran, I know like throughout life, right, we know that the interpretation of letters in DNA is pretty common amongst basically everything. If I see GAT, I'm going to use that information to add a specific amino acid if it had been transcribed, right? So DNA isn't just a building material, right? It's primarily in the real world of life used as information. So is there any sort of concern that you install this sensor into a cell, and then the cell, instead of using it the way you want it to, is going to be like, ooh, GAT, I should, translate this? Are there ways of sort of making sure that your semiconductors aren't accidentally being used as information for building?
Yeah, so usually to transcribe a protein, you need 3 building blocks at a time, so codons. But the DNA that we work with is really so short. It's like 20 building blocks or 25 at most. So it's not really as information technically. You can't really spell out a word with it to be honest. Yeah, so that's really how I would approach that challenge. Yeah.
Not a problem.
Not a problem.
Figured it out.
Solved.
Yeah.
So what are some of like the main challenges of biologically? Like are you have you seen it, or are you concerned that an organism is going to detect that's not supposed to be here? Boot, kick it out of the membrane. Like what are the main challenges?
So one of the challenges is, how do these building blocks come together? So because we're using the approach of creating or synthesizing this DNA one block at a time, the building blocks also are in the same fashion. So one of the approaches is to add these building blocks that are functional or semiconducting or conducting, with punctuated by these phosphodiester's or phosphate links. What this essentially means is that just as you have these negative charges all along the DNA that have to be balanced by something positively charged, so it's neutral, you have to balance the negative charge of the polymer, the DNA hybrids, with positively charged cations. Cations are basically just atoms from which you've removed an electron, so they're now positively charged. In our body, the extracellular or intracellular magnesium or calcium or sodium concentration is pretty low, as Katie would know, but the concentrations that we work with are much higher. So one of the active challenges in using these self-assembled or these nanostructures that we make from these DNA hybrids to either serve as nanocarriers for therapeutic DNA or nucleic acids to get them through the cell membrane, or to have them basically installed into the cell membrane is how do we overcome the negative charge repulsion between these individual chains of these hybrid polymers? And if you use too high a magnesium concentration, that's just not going to cut it with the cells. The cells are not going to be too happy with it.
They'll kick you out.
Yeah.
And we can't just stick some protons in there and call it a day. Like you can't completely negate the negative charge beforehand. You have to do it in the cell.
Yeah. So you can. One of the approaches is to use a higher concentration when doing the self-assembly. That's the correct phrase. And then lower it systematically by either forming permanent bonds between the individual polymers as part of the nanostructure, or reversible connections or interaction. And that would basically stabilize the structure enough to allow you to decrease the magnesium concentration. So that's also one of the strategies that we look at. So that DNA has this complementarity, but it's not just complementarity to within the bases. You can also have small molecules such as melamine or cyanuric acids that are actually complementary to individual bases. And then you can also use these as sort of destabilizing blocks to form these weak interactions that are reversible between the DNA, so that you minimize the need for magnesium. But this is actually an active challenge in the field.
Yeah, it sounds like there's a bunch of challenges that all need to be balanced for this delicate machine to all work out, which sounds like why you would need 3 groups working on the outside, the interface, and the inside. That's really cool. Yeah. So you've mentioned some of the applications for this kind of technique: sensing DNA, viruses, mRNA delivery. What are some other applications that maybe aren't being worked on, but you could see being used for?
So one of the things that came out recently, I think it was published last week in the journal Nature, was a group in Germany created basically a molecular Brownian ratchet using just DNA origami. So what that essentially is they use the DNA as origami as a building block to create basically a 3-dimensional base, and then a small portion, a piece above it, and then it had a hinge within it, and then there was a long arm that would be driven by this stochastic movement of molecules.
Just the random movement.
Yeah, Brownian motion. But because of the way it was positioned on the platform, it would only be allowed to move in a certain direction. And they were able to detect this by positioning certain fluorescent dyes at different positions on their structure. And then they were able to basically wind up a spring, which the spring is actually just a single strand of DNA, by connecting it to the arm. It's really fascinating. They've made this molecular machine that is a precursor of a soft or biological robotic system.
Whoa.
Using just DNA.
DNA bots coming.
Watch out.
We were worried about the robots up here, but what about the robots inside?
Well, I mean, in a way, viruses are kind of like that, right?
Right.
I had to teach myself some basic biology when I started my PhD. So before I started out, I didn't even know that, DNA could be used as a non-biological nano construct, as a nanostructure building material.
Yeah,
And
that's
that's what
a
actually,
physical thing.
exactly, yeah. That's what actually drew me to this group.
Crazy.
We're taking biology and doing engineering with it. I love it.
Yeah.
So you mentioned someone made a smiley face with the DNA nanostructures.
Well, slimy face. Smiley
Slimy
Wow.
face.
Smiley face is very rudimentary. They've also made a nanoscale Mona Lisa.
Whoa.
Wow. I was going to ask how many emojis they've made, but a whole Mona Lisa. We're really leveling up.
I mean, it's not a big deal anymore, because every time somebody presents in our group in the Sleeman lab, they always put in, okay, DNA origami is this, and there's always a Mona Lisa up there. So it's for us, it's pretty hackneyed now.
Right.
Would that be the most impressive thing you've seen made in terms of specificity or scale? Has someone built
that ratchet
something...
sounds pretty rad.
The ratchet is ratchet because it was just published last week, and the Mona Lisa is pretty old.
Yeah.
So it's getting more complex with time.
So now, have to Google that.
Now, if you can make a ratchet that's shaped like a Mona Lisa, and then you hit a button and it spins.
Well,
you release the coiled energy.
If you have the grant money and grad students, you can do pretty much anything.
You can do it.
I want a transformer. I want a
DNA... like an Optimus Prime.
Yeah.
DNA Optimus Prime.
What's the biggest one you've seen with the DNA organ? Like, are any like the size you could actually see with the naked eye, or are they all itty bitty still?
I think the largest ones, to the best of my knowledge, are at the micron scale.
Okay.
You still need some sort of a technique such as atomic force microscopy.
Right. That's still pretty big considering DNA.
Although there was this interesting paper where there were scientists, they took like really tiny millimeter or centimeter sized pieces of gel, and they had different colors, right? So, and gels or hydrogels are basically they're chemicals which have a large volume where the water can permeate, but they're flexible. And we use gels for DNA characterization in the lab all the time. But these hydrogels were, I don't remember exactly what polymer they were made of, but they were flexible and they were floating on water. Basically soft gel cubes. And on each cube, they positioned strands of DNA. So a blue cube had a certain sequence of a specific set of sequences of DNA. And then the red cube was complementary to that. And then, based on the complementarity, they were able to self-sort these millimeter-sized cubes after mixing it. So you basically take red cubes, green cubes, blue cubes, yellow cubes, mix them in water, and then they will self-sort because of the DNA interactions.
Wow. That sounds really useful at the buffet line and the dessert table. So you can get all the Jello cubes all sorted out.
You immediately went to Jello cubes.
Well, I'm hungry.
Jello jigglers. Well, they're You're gonna need a lot of them.
Yeah. Well, it'll be scaled up for industrial purposes, I'm sure. Now, Ghufran, we got to ask you. All right, like, how did you get into this? Like, what was the path that you took from little Ghufran doing math, sitting at the table, learning cursive handwriting or whatever it is that we learned? Do people learn that anymore? No, probably not. But yeah, how did you get into DNA machines?
Well, how did I get into DNA specifically and DNA-based polymers? Well, it's not that exciting a story.
Way to sell it.
My undergraduate chemistry supervisor, in whose lab I worked when I was an undergrad, he said when I came to McGill, go say hi to my supervisor. So I went and said hi to his supervisor, who's Dr. Sleeman, and she wasn't taking any students at that time. So I was almost about to join the Perpechka group exclusively, but then Dr. Hanadi spoke about her research, and she said, she asked me, would you like to hear about my research? And I said, yeah, why not? Because I was meeting professors anyway in my first week at McGill. And when she explained her research, how she uses DNA, I was hooked. And I asked her, can I join your group? And she said, okay, well, we have a project that we're working on with the Perpechka group, so you can be co-supervised. So that's how basically I joined this group. Yeah.
Very cool.
Perfect.
So my primary supervisor, because it's never 50-50, my primary supervisor, Dr. Sleeman, is my undergraduate supervisor's supervisor. So yeah.
Your academic grandparent.
Yeah, that's exactly what another peer of mine said.
But it's good to talk to people in your department, other departments. Like, just meet people, listen to what's going on. You never know what's going to spark your imagination. It sounds like it's not something that was like on your radar until you had that conversation.
No, because I did work with these in the Perpetual group. They work on organic synthesis and creating these semiconducting molecules or conducting molecules for electronic devices such as thin film transistors and solar cells and so on. So I had experience with that in my undergrad. I'd worked on thin film transistors. I did some organic synthesis as well. I worked with semiconducting polymers. So I had more experience in that field. But then because of this collaboration, I ended up sort of shifting my focus a bit in that direction towards DNA. My basically arrival into chemistry was pretty long-winded because my undergrad was in chemical engineering. But to be honest, I was never a fan of engineering, and I really wanted to be a scientist because I originally wanted to be a physicist.
Of course, who wouldn't?
Yeah, of course, yeah exactly.
Said the physicist.
Yeah, but then I had to choose chemical engineering as an undergrad because I had limited options, and so I said, okay, well, if I can transition into chemistry by taking a chemistry minor, doing chemistry research, then let's see where that takes us.
Well, it sounds like you're right at the crux of like chemistry, engineering, and physics. All of these things are applied.
Yeah, exactly. Yeah.
Amazing.
So what were you like as a little Ghufran? Were you always into science? Were you a little science nerd with your little chemistry kits?
I actually was not your quintessential tinkering kind of kid. After I quit dreaming about becoming a fighter pilot as a 6-year-old, I wanted to be a scientist because I was inspired by this Pakistani scientist, Abdul Salam, who's a Nobel laureate. He won the 1979 prize with, I think, Weinberg and Sheldon Glashow. I hope I'm pronouncing his name correctly. So I was really inspired because my dad gave me a biography of Abdul Salam as a 7-year-old. So yeah, I really wanted to be a physicist, but a scientist in general. But I always had a more of a philosophical frame of mind, and I realized this much later when I was an engineering undergrad major. I was not enthralled by the idea of doing engineering and basically running a plant or designing a plant because I felt that a lot of the technology was not new when it came to industrial applications. A lot of what we're doing in terms of chemical production dates back to the 50s or 60s. Well, at least that's the perception I had. So I decided to continue towards my dream of becoming a scientist. I just shifted from physics towards chemistry eventually along the line. And I just felt that also I was much better in terms of aptitude at chemistry than I was at physics.
Sure.
And you mentioned you have like a passion for philosophy of science. What started that? What about the philosophy of science interests you?
Yeah, so what's really ironic is that even though I was hating on engineering as an undergrad, and I was such a big fan of science, I never really asked myself, and nobody ever really asked me, what is science?
Yeah.
And so this began after my undergrad. There was a time before coming to Canada while I was just waiting for my study permit and my documents. I really had not a lot to do. I was just basically sitting at home. And that was basically an excellent time for me to just read and just think and philosophize. And that's when I started asking myself questions about, okay, what is science really? But also fundamental questions about existence and reality.
Yeah.
Why do I have the beliefs that I do have? And so that introduced me to the philosophy of science. Do whatever we know through science, is that epistemically unchallengeable? Is it the ultimate truth? Or is this the best way we have of interacting with our natural world? Or what kind of answers can science provide us with? And for me as a Muslim, that was very important, right? Because we live in a world where there seems to be a conflict between religion and science. And I feel that it's led to the creation of a new religion of sorts, which is scientism, a belief that science is the ultimate source of truth and knowledge.
But do you think that's a perception of actual scientists? Because I often feel like that's how science is perceived by people who aren't scientists. Because for me, at least as a biologist who mostly in my work rely on the scientific method, you come up, you look at the world, you ask a question, you guess an idea maybe as to what the answer to that question is, you design an experiment, and then you see whether or not your assumptions were sort of supported. And it sort of operates in this self-editing way that if you find evidence to the contrary, you're like, I guess I was wrong, right? You don't sort of, at least ideally, stick to your assumptions, you're sort of open to being proven wrong, in which case it's not sort of the steadfast belief despite evidence that I think some people perceive it to be.
Yeah, you have a point, but I think it's also important to remember that as scientists, we're also part of the society, right? So whatever happens in society at large, whatever culture that we're living in, it influences us and our perceptions about the sciences in general as well. And like you guys discussed at the beginning, we as scientists, as professionals, we tend to get very specific about one particular thing, which is our area of expertise. But then we're also novices about everything else within the scientific paradigm, right? So we have to ultimately take somebody else's word for what they're telling us in terms of their scientific discoveries, and that somehow can also become the gospel at times.
And I think it's amazing when you go, even when you go into your own field, there's a bunch of evidence, and then people can interpret it in a wide variety of ways.
Correct.
And the idea that a scientist is like, well, this is my interpretation. If you're not well versed in that field, you can take that interpretation of evidence and say, well, a scientist said it, therefore it's, has some practicable value. But of course, it's not necessarily true. It could be true. And so it's a question of like, what is the evidence? What is the claim being made? And like, how strongly can we believe it or not believe it? And then how is that perceived by someone who's not a scientist? This is true or this is not true. And like, okay, well, we have evidence for this and we don't have evidence for this. And then how is someone who's not versed in science able to evaluate any of these claims? It becomes a really challenging environment for anyone to have a conversation of like, all right, well, these are the agreed upon things. Well, no, these are not the agreed upon things. And so there's a whole other conversation, like a meta conversation that has to happen.
Yeah, there's an interesting article I read in the New Yorker a couple of days ago where I don't know if they were exclusively ornithologists, but biologists were trying to identify the evolutionary origin of a specific bird that's native to South America. I think it's called a hoatzin, an Hoatzin. And this is sort of the duck-billed platypus of birds somehow, because it's defying this traditional approach to evolutionary. So evolutionary thinking, as far as I understand it, is in terms of the tree of life, right? And so you have these branches going out from ancestors of organisms. But this hoatzin doesn't seem to fit specifically on one branch or the other. And scientists who've looked at the genetic information from different birds, from different species, they're still not sure how these fit with each other, because there doesn't seem to be a clean and neat tree. And an important question, a philosophical question, was raised in the article: is should we really be thinking in terms of a tree, which is the idea that I believe Darwin actually sketched in his notes, or is it just like a crisscrossing web of branches that merge and then sort of bifurcate and divide further? But because, and this is what the article said, because biologists and students are taught tree thinking basically in terms of evolution, then perhaps it becomes difficult for them to open up to new paradigms.
Absolutely. Like, just think of like Linnaean classification, how we sort of put animals in little boxes based on, kingdom, class, order, all this stuff. That's all a human convention because we like putting things in drawers, right? Look at our house, right? We put our sweaters in one drawer.
Speak for yourself.
Vinny just has a pile. Okay, not everyone does this. But right, that's a human thing. That doesn't necessarily represent the natural world, right? And then when we see reality sort of being in conflict with how we like things neat and tidy, our brain sort of goes on the fritz.
Yeah. I mean, there's also the debate of what we perceive of in terms of the natural worlds. Do we look at it as, I'm not a philosopher, but my understanding is that you can either be a realist or an instrumentalist about theories of science. So what we observe and we look at, is that really something that exists? Or is the instrumentation that we're using or our approaches and theories, are they sort of the best possible way to explain this given our limited knowledge and the limited number of interpretations that we're employing currently?
Yeah, and I think I think science is making guesses, and we're making it, if you can make a prediction off of your model, then that feels like a workable model. And the moment you can't make a prediction off of it, then you have to alter your model. And I think that's the leading edge of like, all right, well, now this is a reliable prediction. What else can we predict? And like the work that you're doing, you're making like all of these, you're like, oh, if we try this and we try that and it should work and it does work, well, then you have a good model, you know. And like science is always testing its model and looking for places where models fail. And that's where the exciting part is, like, oh, our model doesn't work anymore. What's going on there? And so now we have to figure out what the next step is to make our models even better. But at the best, we only have models. We don't have truth, quote unquote.
Yeah, and exactly. And this ties into the fundamental problem that's tied with the assumptions underlying the scientific method, which is that we have to assume in order to do science that the natural world is uniform, right? So whenever we say that adenine is complementary to thymine, right? So this is based on prior experience, and so we have to assume that the natural world works in an organized way, and that there is a continuity, right? But this, so for instance, we know almost with certainty that the sun is going to rise in the east tomorrow, but we can never be fully certain, and this is the black swan problem, or the problem of induction. That, you observe 1,000 white swans. You can be fairly confident it's the next swan is going to be white, but you can never be certain.
Yeah. Always induction.
Yeah.
Well, we could talk about philosophy science for another hour. I wish we had more time. But before we let you go, Ghufran, I've got to ask you. You mentioned being a squirrel paparazzo and being a McGill student. I'm going to make an assumption that your favorite subject is cloudberry. A, is that correct?
Oh, yeah.
B, tell us about cloudberry for those of you out there who are unfamiliar.
Yeah, for those of you who are unfamiliar, there's a white squirrel that lives on a tree behind the Audemars Chemistry Building at McGill. And according to Reddit, the McGill Reddit page, it's called cloudberry. I don't call her that. It's just something that's popularized by the students of McGill online. But yeah, this is a squirrel that you will occasionally find foraging through the trees behind the chemistry building, or in front of the conservatory, which I think is about to shut down. Yeah, it's beautiful. It's gorgeous. Which is why I'm not your run-of-the-mill squirrel or wildlife photographer. I'm a paparazzi because this squirrel is a celebrity.
I gotta ask you though, how sure can you be there's only one?
Yeah, that's another sort of online debate. Some students think that there are multiple.
The prestige.
Yeah, I just feel that there is just one. Firstly, because this is a pretty rare occurrence. You don't normally find leucistic animals, so it's not an albino. I think it's a leucistic animal. You don't normally find these as commonly, so probabilistically, of course, we can't be certain. I think this is the only squirrel that's leucistic, that's purely white, that's out there. Although I have spotted a squirrel that's partially white. So it might be her offspring.
Wow.
Oh, to be continued.
To be continued. So we can rename the black swan problem the white squirrel problem.
Yes.
Yeah.
Well, Ghufran, this has been fantastic.
Amazing.
Thank you so much for sharing your research. It sounds super cool, and we wish you the best of luck in your future.
Thank you so much for inviting me. It was a pleasure.
Awesome.
Great.
Wow.
What a frontier.
It feels like we accidentally found someone from 100 years from now.
Yeah, but now.
Or deliberately found.
It's the stuff that people are doing these days just kind of boggles. Like unimagined. Like we went from discovering DNA. What year was that? Like the 50s? Like the helix.
Understanding the physical structure was in the 50s.
The 50s.
I believe.
Yeah. So like 70 years later, we're now using it to build stuff. Like going from stone tools to like building buildings out of it. It's wild.
It's crazy. And yeah, what else?
What's happening 70 years from now?
I don't know.
I don't know.
But it's true. This season of Learn Real Good in particular, there's been a lot of sort of futurism.
Yeah,
with a lot of artificial intelligence.
But it's not even futurism. It's now. It's like what you imagine to be happening. Like, oh, we can imagine a future. It's like, no, they're doing it. Like people are working on these problems that seem like science fiction to me.
7 years ago, I had a bunch of Rubbermaids on the roof of the Stewart Biology Building at McGill doing an experiment.
Yeah. Wild.
Maybe I was just living in the past.
That's okay.
Well, Vinny, we learned a lot today.
I learned so much.
We learned about how to optimize boiling of water.
The key problem.
We learned about voles fighting back against butcher
Vole
birds.
versus Shrike the movie. Excellent.
Coming to theaters this fall. And of course, tons about DNA, using it as a building material.
Yeah, and DNA engineering.
Thanks to Ghufran.
Thanks, Ghufran.
That's wild. Well, I think we're wrapping up.
That's our episode. If you're interested in following us on social media, check us out at LRG Pod on Facebook, Twitter, Instagram, and TikTok. That's right, TikTok. For you oldies out there, we're on there. And finally, if you're interested in joining us as a guest, we're always looking for new people. If you're a grad student in STEM and you want to join us, send us an email at learnrealgoodpodcast@gmail.com. learnrealgoodpodcast@gmail.com.
And if you're a fan of the show, we would love if you could take 5 seconds and write a review. We have a few 5-star ratings. Thank you, folks, for doing that. We have hundreds of downloads each week, and yet 1 written review.
If you're looking to support us, we'd really appreciate a great review.
Yeah, even an okay review.
Even an okay review, but preferably a good review.
Yeah, if you don't like the show, maybe
keep that to yourself.
Keep that to yourself.
Keep that to yourself. Put that in your pocket. Write it in your diary. Yeah, write in your diary. That's a great place to vent.
Your live
I'm
journal.
Sorry. What? We have not yet figured out TikTok, but we've got live journal on lock.
All right, that's enough from us today. Thank you for listening.
Thank you so much. We'll see you next time.
Thank you, Ghufran. Thank you, Vinny.
Thank you, Katie.
Bye-bye.
Bye-bye.